Good morning, afternoon, and evening to everyone joining us. Please bear with us for a few minutes while we wait for the attendees to flow in. Okay, a quick housekeeping rule. Please post any questions that you may have in the Q&A tab, and it will be addressed during the Q&A session. Access to the recording of this webinar will be emailed over to your registered email address within 48 hours post-event. On behalf of Vizgen and Global Engage, I would like to welcome you to today's webinar entitled Pioneering the Next Era of Spatial Biology with MERFISH 2.0 and MERSCOPE Ultra Platform. I would like to thank Vizgen for sponsoring this event. Our presenters for today are Alex Genshaft, Product Manager for Reagents and Consumables, Vizgen, and Professor Mark Watson, Margaret Gladys Smith, Professor in Pathology and Immunology, WashU Medicine. So, to kick off today's session, I'd like to invite Alex to take on the virtual stage to deliver his first presentation of the day. Over to you, Alex.
Thank you, Kamini, for inviting us and having us on this awesome stage. Can you have my slides?
Yes, we can.
Great. So thank you all today for joining us. I'm really excited to be introducing you all to MERFISH 2.0, which is introducing the next era of spatial biology. So how to really keep pushing this field further and further? Spatial biology has been gaining traction over the last 5-10 years, with global grants going up and up and up. And we've identified key research areas where these are performing incredibly well in developing new and novel insights across neuroscience, immunology, and oncology. But there are challenges that remain. So sensitivity is a big one, sample compatibility, especially FFPE archival samples, and then maintaining that transcriptomic integrity throughout all of these processes is quite key to generating high quality data. MERFISH 2.0 is really addressing these all head on, enabling us to map challenging tissues with high dynamic range transcript capture. And from all of those transcripts getting for each cell, more of our cells are going to pass through filters. From those more cells, we're going to generate more insights. Those insights are going to come through identifying key types and states across various abundances from highly populated to very rare cell types. And from all of those cell types that we've now annotated, we can dive deeper into transcripts that they're expressing and cover more spatial interaction, neighborhood analyses. And now MERFISH 2.0 is really built on this incredible legacy of MERFISH 1. It's the same trusted technology, just better and optimized. We're able to get more transcripts, as I was just saying, which are enabling us to chart with high reproducibility and high dynamic range all of the transcripts that are expressed in our tissue. And from that expression, we're going to get more cells, which will enable us to explore the macro and microenvironments of those tissues. And in this cell type, spatially resolved cell typing, and the enhanced sensitivity and specificity to really drive your insights. And those insights are going to come from mapping biomarkers, signal pathways, rare cell type, cell interactions, and all with the uncompromising subcellular transcriptome resolution that MERFISH is known to provide. Now, MERFISH is the leading cell resolution high-plex in situ spatial transcriptomics technology. We've over 300 published papers and preprints between MERFISH and MERSCOPE, and this number is growing at a staggering rate annually. The science behind 2.0 is called MERFISH. It stands for multiplex error robust fluorescence in situ hybridization. And from that, we're going to do combinatorial labeling. So we're going to tile across transcripts and then bring out probes in a sequential fashion that will light up these transcripts one by one. From that barcoding, we're able to decode which transcripts are expressed in which subcellular localization. Diving a little bit deeper into the molecular of this, If we look at the top left, we see what happens when we have a high-quality transcript and a high-quality sample. We're able to tile probes all the way across the transcript, and then we can bring in our readout probes, which are these blue and red icons, and those will bind and generate a fluorescent signal. Because we have the sum of all of these readout probes, we get a very bright signal, and we're able to detect that transcript accurately. When we have a low quality sample, there are fewer encoding probes bound, and that leads to fewer readout probes bound. And because of that, the signal is decreased and we have more difficulty calling those transcripts. The MERFISH 2.0 workflow really optimizes for this by beginning to build trees of binding sites off of these encoding probes. So you can see that in the bottom, we have the same number of encoding probes bound, but we're starting to approach the readout probe density of the high quality sample. And this is, of course, just a schematic. So this is not the exact enhancement amplification that is present. It just gives you a hint of how we're delivering on this higher sensitivity. And it's really coming together from the RNA anchoring. So we're going to tile across that transcript and anchor it into our gels. And we've optimized the probe binding structure so that there are higher occupancy rates on both the encoding probe side and on the readout probe side. And then we've further augmented that readout probe structure to increase and amplify the fluorescent signal that's being generated. So this is all leading to some serious key benefits from MERFISH 2.0. We're able to map these challenging tissues across FFPE and archival tissues, generating that high quality, highly reproducible spatial data. Getting improved transcript capture and uncompromised subcellular resolution with this high dynamic range. And as I was saying before, all of this is leading to more cells, which are leading to more insights. With the richer data sets from more cells passing filter, which is enabled by the higher transcript detection personnel, we can identify cell types and states, whether rare or abundant, and uncover more spatial interactions between molecular signaling pathways and cellular crosstalks that are beginning to inform disease and therapy research. So we've done a ton of validation of this in-house, and we've seen a tremendous improvement, up to 8-fold sensitivity increase with MERFISH 2.0 compared to MERFISH 1.0. And I'll draw your attention to just a single data point here. If we look at the breast cancer tissue array core 1, we're specifically going from a data set that is unanalyzable with MERFISH 1.0 data and bringing it to an analyzable state with MERFISH 2.0. And that's just one example. You can see across both of these plots, both in human and in mouse tissues, we're demonstrating a great improvement across all of these different tissue types. Now, improvement without reproducibility is not necessarily an improvement. So we're able to show on the left here that MERFISH 1 and MERFISH 2.0 counts are highly, highly correlated, with, of course, a drift above the y=x line, indicating that MERFISH 2 is generating more counts. And this is highly reproducible against RNA sequencing as well. So we can look at brain, breast, lung cancers, and breast cancers in the human tissues, and spleen, heart, lung, small intestine, brain, it's fixed, frozen, or fresh, kidney and liver, and we see, again, highly reproducible correlations here. Now, we validated this across multiple sample types, a sample which are colon cancer, human breast cancer, human brain, and mouse brain. And performance is really driving biological insight. So we've demonstrated this performance across a variety of tissues. We've shown it in archival and fresh frozen, and it's generating robust data, again, highlighting that previously unusable breast care core that we could not generate data from initially, that we were then able to generate data from. And this increased sensitivity is going to lead to confident identification of cell types and better analysis of the interaction. And MERFISH 2.0 pairs especially well with the MERSCOPE Ultra, which is enabling us to dive deeper into spatial biology, higher throughput, and higher resolution than ever before. This really brings us to the ultimate spatial biology ecosystem, between our sample prep protocols with MERFISH 2.0, data acquisition with MERSCOPE Ultra, and our data analysis and visualization with the visualizer software. Now, we'd love to explore this with you either in your lab or in our lab or with a certified service provider. We have a ton of options for getting you up and running and generating high quality MERFISH data. So how do we get started? We'll start with choosing your solution, as I just mentioned, between your lab, our lab services, or a certified service provider. We'll move on to choose a panel, whether it's pre-designed panels or custom design, and we'll help guide you through this all every step of the way. And our promise to all of you, all of our customers, is that we will continuously innovate. We'll be developing new products, optimized protocols, and further guidance for any tissue types. We're delivering this all with a white glove service across many field teams, from sales and field service to tech support, R&D, and customer support, and all of the internal teams that we bring to drive your success. And we are committed to scientific partnership. This is not a one-way street, and we are here to help design studies, problem solve, and more. So we're really forward to it, and we hope that you reach out. I want to thank you for your time. I know it was a quick talk from me. My name is Alex S. Genshaft. I'm at Vizgen. You can reach me by alex.genshaft@vizgen.com, or you can write in straight to info@vizgen.com. Thank you all.
Thank you for that great presentation, Alex. I would like to introduce our next presenter, Professor Mark Watson. Mark, the room is now yours.
Okay, I think, can we unshare?
Yes, Alex, if you can. Yeah, that would be great. Thank you.
Perfect. Okay, can everybody see my slides okay?
Yes, we can.
Great, thanks. Well, first I want to thank everybody for inviting me to give this talk today. We were very early adopters of MERFISH technology. We originally used it actually for a lot of our colleagues in frozen mouse brain tissue, where it worked quite nicely. My own background and other faculty in our department are very much interested in paraffin fixed sections that we use in routine clinical practice. And so we're particularly interested in using this technology platform on that type of material. And so we were very excited and feel very privileged to test, beta test the MERFISH 2.0 specifically for formalin fixed tissue. And as you'll see here, kind of in a real world example with our beta testing, it's working quite well, and we're able to, I think as Alex said, really move our research forward. First, I want to thank a number of people. My clinical colleague, Dr. Rebecca Aft, who is a clinical breast surgeon oncologist, and is working with me to really get these specimens and ask the clinical questions that are so important. Dr. Richard Cody, our previous department chair, who was instrumental in getting this platform into our department initially. And then really a great technical team, Jackie, Desiree, and Karina. Jackie has worked through step by step all the chemistries involved in getting this to work optimally on formalin fixed tissue, where not only the tissue processing, but the histology that's done is really critical to getting the kind of results that we see here. Our informatics group, Madarima and Wenji, who are using tools that are available, scripts, but also developing new methods to take these new types of data sets and really ask probing clinical questions. Lastly, I'd have to say that, you know, the next best thing after MERFISH 2.0 at Vizgen is Shayak Mitter. He has been really instrumental in helping us get things working. He's an incredible technical resource. If you haven't worked with him, one of the things we really appreciate about working with Vizgen, as Alex has alluded to, is really the collaborative efforts that go on in optimizing this for our science. And also Miguel, who really was the first person to get us hooked and using this platform. So thanks again for everybody and Vizgen for inviting us to talk. So our research focus is on triple-negative breast cancer. If you're not familiar with the disease, it's a clinically aggressive type of breast cancer. It actually has an increasing prevalence in young women. And even in the presence of neoadjuvant therapy, about 40% of those patients do not have a pathologic complete response. So it's a very important clinical issue. And in the absence of this primary response, many of these patients frequently go on to metastatic progression. Oftentimes that's to the central nervous system where treatment options are very limited. So a number of important clinical questions and things to figure out about who are the patients that are progressing, how do we prevent that progression and increase survival in this patient population. Biologically, triple-negative breast cancer in itself is very heterogeneous cellularly. It's often very poorly differentiated. It's just a mass of cells, as you'll see. So it's really important to understand the underlying molecular and cellular heterogeneity too. And while, you know, in the past decade, we might grind up tumors and do bulk RNA-seq, and maybe now we do single-cell RNA sequencing from dissociated cells in tumors, really the ability to take clinical specimens and look at them histologically and molecularly has transformed the way we can understand these tumors, and particularly for us, understand what are the events that are happening to promote early dissemination of tumor cells. So this is sort of our experimental paradigm where we've initially started to look at occult disseminated tumor cells present in the bone marrow and other sanctuary sites. This is a problem involving looking for rare tumor cell detection. It's not spatial in so much as that we're getting aspirates of cells, but it is trying to detect and classify rare tumor cells in a sea of other immune cells, which are obviously important, but not our immediate focus of attention. But more recently, because of the technologies that are now available, we've gone back and looked at the primary tumor and are asking now histological and spatial questions about what are the features, the organizational features that promote early tumor cell dissemination and eventually metastasis to the brain, which ultimately is something that we're trying to prevent by recognizing these events early, understanding their underlying molecular mechanisms, and seeking ways to prevent that process from going forward. Now, as it turns out, we're using MERFISH technology in both of these areas. What I won't talk about a lot today, but I'll show you one slide of how we're using MERFISH to essentially perform molecular cytology for very rare cell detection and classification. And really the rest of my time here I'll spend on the beta testing that we've done with MERFISH 2.0, looking at the spatial transcriptomics of the primary tumors themselves. So along with Dr. Aft, we've developed a clinical paradigm where we can look at consented patients with triple-negative breast cancer during their standard of care. We're collecting not only the primary tumor tissue specimens, but also bone marrow aspirates at newly diagnosis, after neoadjuvant therapy, and at 1-year follow-up. So we can look dynamically both at the primary tumor itself while it still exists and these sort of reservoir sites of disseminated tumor cells to understand what's going on over time. We actually use a technology that performs unbiased cellular enrichment for the bone marrow mononuclear cells, which are also the components that harbor the disseminated tumor cells. And up until recently, we had sort of been performing conventional single-cell RNA sequencing. But applying MERFISH to this has really changed the way we're doing this. And so we can take a custom-designed 140-gene panel that we've curated through the literature to know the key sort of signaling molecules involved in tumor cell dissemination, and we can place about 200 to 500,000 individual cells on the disk and look at expression. And by designing the panel, we can generate data like this, where we're looking at a high-power view of one of these bone marrow aspirates. We're looking across cells, and it's very easy to pick out here, based upon the pattern of gene expression, those cells that are disseminated tumor cells, DTCs there, versus the other cells in the bone marrow, lymphoid lineages, and others like that. So it's a very nice, high sensitivity, high power way to look for these rare cells. I will say that this is from MERFISH 1 chemistry, so we haven't applied MERFISH 2 yet, but we're really excited about as I'll show you in a minute, the advantages of a signal in MERFISH 2, I think it's really going to up this. And as you can kind of see here, we're looking at over 500,000 cells with our panel. And if we're looking for very rare cells, here's a high power view looking across a sea of cells there. You can almost see like starry sky, pick out those individual cells that are the tumor cells in the background of all these normal bone marrow cell components. So the idea that rare cell detection, either whether we're doing it here or whether we're doing it in the context of a tissue spatially, I think is very, very powerful methodology. And although this is not spatial per se, we're looking at 200 to 500,000 cells at a time. If we were doing this by single cell sequencing, generally we're only looking at about 10,000 cells. So our ability to look at many, many more cells is great. The other advantage here is that we're not losing any cells in library preparation or sequencing. We're looking at every single cell in the sample, which was always a problem when we were doing this approach using single cell sequencing. And unlike flow cytometry, where we are novices at it, but at best we maybe do 6 or 10 antibodies. We need to have antibodies here. We can engineer 140 to really now up to 1,000 RNA targets to look for rare tumor cells, rare other cells, and classify them. And again, because this is all on a single cell data platform, it's computationally very friendly. So everything that we could do in a single cell RNA-seq experiment, we can do with this data in terms of our workflows and visualization. So this is sort of an aside, but we're really excited about using this technology for this type of problem, in addition to the spatial questions that I'm going to talk to you about now. So molecular profiling in formalin-fixed tissue has always been a problem for us, but it is just tremendous advantages over getting frozen tissue. I run a biobank where we've got thousands of frozen tumor tissues, but they don't have the type of depth of variety, site, and clinical question that can be asked. So by able to access formalin-fixed tissues, we can look at clinical specimens, we can look at clinical trial specimens, and that gives us access to diagnostic biopsies, primary tumors, recurrent lesions, metastatic lesions. All of these have deep annotation, and they allow us to address some of the complex clinical questions that we just can't do if we're stuck with frozen tissue. The other thing about FFPE tissue is that we have a lot of histological detail. This is particularly important for one of our research aims, where we're trying to use AI-enabled histological evaluation of tumor tissues along with underlying molecular profiling. So the ability to actually use this tissue that's amenable to both type of formats is really, really a plus for us. Obviously, formalin-fixed tissues are stable, they're easier to process, and as Alex has alluded to, excuse me, as Alex has alluded to, you can do things like tissue microarrays. And so we've actually, as Alex has shown, done some MERFISH on triple-negative breast cancer tissue microarrays. This, again, is using MERFISH 1, so we're really excited about using MERFISH 2 chemistry. You can see here kind of at a high-power view that the quality of the tissue can vary from spot to spot, but we're really excited about applying this type of method now to MERFISH 2. The problem, of course, with formalin-fixed tissue is that RNA degradation can be significant. It's very tissue dependent. The clinical site where you get the tissue block is a variable, and how that tissue was fixed, whether it was fixed on a Monday or a Friday, can make a big difference as well. And so, for example, in a project we did for RNA sequencing, looking across 4,000 tissue blocks and measuring RNA quality by DV200, this is sort of a typical distribution that we see. Our sweet spot for good RNA sequencing is around a DV200 of about 28. Above that, we're reasonably good. We're reasonably sure we have good data. Below that, it's hit or miss. And so, as you can see, if you look across blocks, there's quite a range here. I'll show you that in our pilot studies, we're quite fortunate to pick blocks that were somewhere in the DV200 of 30 to 40 range. So actually a little bit better than this normal distribution, but the ability to do this types of analysis on fixed tissue is very, very powerful. So I think this is the one slide maybe you should wake up for. This is similar to what Alex has shown, but this is actually in our hands with Jackie and Karina and Desiree cutting real tissue in our lab. So we took one of our triple-negative breast cancer specimens here. Usually we do sort of running cuts and isolate RNA and check the quality. This particular sample had a DV200 of around 37. And we use serial sections to perform analysis using the original MERFISH chemistry and MERFISH 2.0 chemistry. This is sort of the metrics we saw, and generally we would see for FFPE using the original MERFISH chemistry, a number of metrics there, but I think the one that is probably most to pay attention to is the median transcripts per cell, about 61. When that same section was done with MERFISH 2, you can see now here 5 and sometimes 6-fold higher transcript detection, so considerably more sensitivity. And this actually for us translates into being able to pick up more transcripts and really making more biological insights because of that. I'll also say the other big advantage of 2.0 is some of the other features, the software features. So the 2.0 software and upgrades features a HTML report. If you've ever done 10x sequencing and run Cell Ranger, it's sort of a similar type of highly portable HTML report that gives you QA metrics. It runs basic clustering to give you a spatial and UMAP and even dot plots of gene expression, count cells, the transcripts, and the stains. So in a facility where we're actually running things for other people, it's a really nice way to give the user just sort of an immediate glance of what the data looks like. If they're not tied to a bioinformatician, it's easy to open and interpret and see a lot of data a lot of ways very quickly. The visualizer software itself, I'll show in a minute, has, I think, really improved performance. It just runs better. It has more features. It's a little more usable. So not only is the chemistry much better, but the underlying software to do the analysis, even if you're not running your own scripts, is much, much better and has really accelerated our ability to analyze these samples. So again, our focus has been on looking at how signaling molecules, pathways associated with disseminated tumor cells in triple-negative breast cancer are organized regionally in the primary tumor, and can we in fact understand patterns of cellular and molecular signaling in the primary tumor that predict early dissemination. So in this sort of beta testing pilot, we took a number of our triple-negative breast cancer primary tumors. These are patients who received neoadjuvant therapy. Despite that therapy, however, they did not have a pathological response. And so we're looking now at the primary tumors after either chemotherapy or immunochemotherapy. They have not responded, and we're asking what's going on in these tumors that they have not responded, and importantly, do we see any early signs that tumor cells are disseminating from these to distant metastatic sites? So this is a typical TNBC sample that we did. For the pilot study, we were using the 500 gene oncology panel, the out-of-the-box panel, which is great, has a number of very relevant genes, many that overlap with our DTC-associated gene panel of interest. Here you can see Monterima and Wenji have just run a basic clustering, lighting clustering, and shown the regional expression of these genes. I think the thing that's remarkable here is that you see in many of these tumors really different regional patterns of expression. These are all almost pure tumors. So what we're looking at here is not only different cell types, but we're looking at tumor cells that have very different programs of gene expression. And so we can look here and begin to see these different and complementary patterns of gene expression, looking at EGFR, which is frequently expressed in TNBC tumor cells themselves, and then two molecules that we're very interested in, secreted Frizzle-related proteins 1 and 2. These are proteins that drive tumor cells and complementary drive cancer-associated fibroblasts, which promote survival and dissemination of those tumor cells. And here, it's very easy to see on sort of a one-off gene expression map how there are complementary patterns of expression in SFRP1 and SFRP2 compared to the primary tumors. And if we look across different tumors, here's another patient, similar therapy-resistant tumor that was resected. Again, this is almost pure tumor with intervening stromal and immune elements, but you can see even within the tumor cells, very regional differences there. If you look to the west, you can see a population of tumor cells that clearly have a different expression program than the other cells within the bulk of the tumor. And again, we can kind of look at these same genes. We see the same pattern of complementary expression between the SFRP1 and SFRP2, underlying sort of the tumor cell populations, and then the underlying supporting cancer-associated fibroblasts. Interestingly, patient number 3, this is a patient who had a partial response to therapy, another triple-negative breast cancer. In this particular patient, there's lower tumor cellularity, but a lot of infiltrating lymphocytes, a lot of stroma. And here we actually see an opposite pattern between the SFRP2 and 1 in terms of their arrangements of cells and interactions. And so beginning at least on a single gene basis to understand how these different patterns of expression correlate with the clinical behavior of the tumor. Obviously, it's a complex interaction between the SFRP1, SFRP2, other signaling of Wnt. An example of a complexome is shown there in the bottom corner. But the ability to look at many of these genes, all of or most of which are in this 500 gene panel, is going to allow us to sort of reconstruct this type of diagram, but in the histological context of these tumors. Now, many of the clinical samples, at least in breast cancer, tend to be diagnostic biopsies on initial diagnosis. And so the tissue specimens often look like this. This is a needle core biopsy from a triple-negative breast cancer patient before therapy. There's a lot less tumor cellularity here. The other advantage, however, is that these pieces of tumor tend to be smaller, so the ability to actually put several patient biopsies on a single slide, although that involves some intricate histology, is very doable and allows us to compare patients, perhaps responders versus non-responders, all on the same slide. I think if you're adopting in particular the MERSCOPE Ultra, it even gives you some more space to do that. So beyond doing tissue microarrays, you can also do sort of multiple specimens across a cohort of patients, which makes things very efficient. Here again, in this particular case, you can see, again, the cell clustering, a combination of tumor cells that are expressing EGFR and MUC1, and then the complementary, again, the SFRP2 being expressed in the cancer-associated fibroblasts around this tissue as well. And then just another example of how now we can go from single gene level to more complex analysis. This, again, is that case of a TNBC that was partially responsive to immunochemotherapy. Again, using the 500 gene oncology panel, we can run scripts to create clusters and map those. But I think the other advantage of the MERFISH is really, again, the software tool where you can use the Visualizer 2.0 and get a much more interactive, much more detailed feel for what's going on at the individual transcript level, but also at the cell cluster level. And of course, the Visualizer tool, being essentially a virtual molecular microscope, allows you to zoom on in to look at cell populations that it's not so easy to do if you're just running Python scripts. It's very interactive. The other thing, however, is that visually it looks cool, it's very pretty, but it's hard to do anything computationally with. But because, again, this is essentially single cell data, all of our pipelines that we use for single cell RNA sequencing are amenable to this type of data. And we can begin to computationally do things like look at spatial neighborhoods and identify clusters of cells that tend to be associated with each other. And then obviously looking at the underlying gene expression in these different clusters to understand, what groups of genes are being expressed spatially in those different organized clusters. Lastly, coming back to our paradigm where Dr. Aft has patients that are getting diagnostic biopsies. Sometimes they're getting lymph node biopsies if they have lymph node involvement. They're receiving a neoadjuvant therapy. And again, we're focused on those patients with triple-negative breast cancer who have not responded to therapy and asking what's going on in those samples. And so we can also obtain FFPE tissues from the surgical resections. We can take an example here of a patient who had both the diagnostic core biopsies and the surgical resection. Again, we took these samples side by side and applied the 500 gene oncology panel. Here again, I'm showing you the one below again, and here's the corresponding biopsy from that patient. But importantly, we've been trying to work out how we can reconcile these two different pieces of tissue, one which is just a core biopsy of a sampling, one which is the entire resection specimen itself. At these very high-level views, it's kind of hard to understand what's going on, but having single-cell spatial resolution to these, we can begin to look at spatial neighborhood analyses, and we can see changes in those cell-cell interactions at the cell-to-cell level, and begin to correlate those patterns of changes we see with whether or not these patients are going on to create tumor dissemination, either occultly in bone marrow or clinically obvious metastases to other visceral or central nervous system sites. So our pilot is fairly much complete with the samples that we looked at. Obviously, ongoing studies are to get additional samples, continue to optimize, improve processing. I think Jackie could tell you it's very tissue specific. There's a little bit of trial and error, but particularly with the new MERFISH 2 chemistry amplifying signal, as I've mentioned, we've been much more successful in looking at FFPE tissues. We're very interested in working with Vizgen. Again, they were very collaborative in developing tools to co-register the really high detailed H&E images that we have of all of these with the spatial data. And then the 500 gene oncology panel was a great out-of-the-box solution to test the chemistry and look at a lot of insights into the cancer cells. But again, I think the other advantage for us is the ability to customize probe sets. So using reference gene sets in breast cancer, asking specific biological questions, we'll be able to design our own custom probe sets that will really be able to interrogate more finely the biological processes that we're interested in. And finally, the visualizer tool is fantastic for looking at the cells and the tissue itself. And Mariam Wenji and myself are working on other methods to sort of normalize and perform comparative neighborhood analyses. Again, very much interested in changes in the molecular histology of what's going on in these tumors over time. Okay, so I'll stop there, and I guess we can take any questions or discussion.
Thank you, Mark. So we will now start with the Q&A session, and I would like to start with the first question. Let me look at the questions that we have. Okay, so we have one. Can our current MERFISH upgrade to MERFISH Ultra, or we have to get a new machine of MERFISH Ultra? And I think Alex would like to answer this question.
Thanks. It's a great question. So the MERSCOPE Ultra is an entirely new piece of hardware. So you will need to upgrade that with a new platform. And we're committed to working with all of you to enable that. So please feel free to reach out to me or your local Vizgen rep, and we can getting you upgraded from the MERSCOPE to the MERSCOPE Ultra. We would love to support that.
Okay, great. Next is, what are the differences between MERFISH 1.0 and 2.0 outputs?
Yeah, this is a good question. So Mark actually alluded to this. One of the main improvements we've released with this new version of software is producing an HTML output, which gives a high-powered view of high-level view of how your data performed and how the MERSCOPE performed. You get, as Mark highlighted, cell type clustering and some marker genes that come fresh from that, and provide a great way of assessing how that sample performed. Other than that, And that will come with either MERFISH 1 or MERFISH 2 on our release software. And that there are very minimal updates to the output files, and they should plug and play well with your current workflows.
Okay. The next one, can one combine MERFISH 2.0 with an antibody stain?
Yeah, this is a great question as well. So it's similar to MERFISH 1, we can still employ co-detection with up to six protein markers, and we can layer on top of that our cell boundary stain.
Okay.
So for 9 markers.
And we have a question from Isaiah. How difficult, or generally speaking, how is co-registration of H&E image with MERFISH 2.0 results is done?
So I can say right now we're doing it visually, and visually it sort of depends upon the geometry of the tissue section. You know, we cut serial sections, we try to orient them in the same way. So at the moment it's being done visually, but we're actually looking into ways to do that informatically so that you know, at the cell coordinate level, you could co-register those things. And I don't know if Alex has more to say about that, but that's something we're very interested in doing. I didn't show a lot of high power images, but you can find, you know, with these 40x scans that we do, you can find co-registered cell populations that you see by H&E, and then look at the corresponding, you know, molecular features in that same region.
Thank you so much,
Yeah.
Mark.
It's
Yes,
definitely
you have
something
anything
we're
to add,
continuing
Alex?
Sorry. Yeah, it's definitely something we're continuing to explore at Vizgen. We know it's of a lot of utility to pair and match with other gold standard methods that you have in community. But to Mark's point, it really depends on the quality of the sectioning and making sure that you're going to get those geometries and have them be able to line up.
Okay, just a quick announcement. I would like to ask everyone to put in your questions into the Q&A tab and not the chat. Otherwise, we might miss it out. Thank you very much, everyone. Okay, moving on. What spatial gene expression signatures are associated with immune evasion or therapeutic resistance in HER2 negative TNBC? And how can they inform treatment strategies? I believe this would be for Mark.
You know, that's a great question for a grant. It's something that we're looking at. You know, I think there have been a number of papers in the past year or two that have used maybe MERFISH, but you know, other spatial technologies to kind of address these subpopulations. Our particular interest is in finding those components that make it tumor cell dissemination permissive. Some of those might be immune, but some of them might be, you know, other factors too. But really great question, and at this very moment, I'm sure there are labs across the country that are working on that.
Okay, great. Next. The 4,000 samples that you've analyzed for DV200, were they all breast tissues?
For that particular study, they were.
Perfect. And how are you looking to design your custom probe sets? Please share some insights for triple negative breast cancer.
Yeah, so, you know, our strategy there, because we were initially focused on disseminated tumor cells, which often have different molecular and therapeutic vulnerabilities compared to primary tumor, we basically used a semi-AI, semi-human literature search for all markers that had some relevance to the biology of DTCs, selected those, and then really pasted that on top of like gene expression, GTEx data to look at tissue specificity, and came up with a list that way. So sort of soft informatic-based approaches to pick those genes that might be expressed most relevantly in the tissues that we're looking at, and might, at least in the literature, have most biological relevance.
Perfect. And we have one more question. Can spatial transcriptomics be integrated into routine pathology flow works to improve diagnostic accuracy or prognostic assessment in HER2 negative TNBC patients?
So I would say the platform itself is probably not ready for clinical laboratory use for a number of reasons. But I think importantly, and you know, our goal is by looking at thousands of transcripts, we can identify, candidate genes that might be more relevant, which could then be translated into more clinically applicable platforms, be it antibodies or even, clinical in situ hybridization.
Perfect. Well, we're getting quite a number of questions. Would it be possible to detect fusion transcripts or mutated transcript with MERFISH 2?
I'll take that one. So it will be a little difficult to detect mutations and fusion transcripts. Depends on how, so on the mutation side of things, on both sides of things, depends on how big the change is. Again, piling probes cross our genes, so we'd need a number of those probes to be distinct between form A and form B, if you will. So with a fusion, it's more likely. With a mutation, a point mutation, it's less likely, but it's definitely an active area of research here at Vizgen. Thanks.
And Isaiah says, add on to that same question, can one custom specific sequence to detect a TRC or BRC-based clone?
So we would be open to looking into that. It will depend on the specific clone and how orthogonal that is going to be against the rest of the transcriptome. Given that we're generally looking for a large number of clones, a large number of probes, it's a little low likelihood at this point, but it's definitely something that we will investigate with you.
Perfect. Next question. Is a MERFISH Ultra instrument required to run MERFISH 2.0? Alex, that's for you.
Rapid fire. No, so MERFISH 2.0 is compatible with either the MERFISH, as Mark was showing today. All of his data was generated on a MERFISH 1 instrument, or MERFISH, excuse me, a MERSCOPE instrument. MERSCOPE Ultra will all be compatible with MERFISH 2.0, but it is not required.
Okay. Next. How many samples have you ran with MERFISH 2.0? That will be for you too, Alex, I believe.
So internally, we've run hundreds of samples. Externally, we've ran approaching 100 samples. I'd love to hear from Mark. How many samples have you ran?
Well, yeah, so for our MERFISH 2 pilot, we did about 18 breast cancers, both biopsies and resections. For MERFISH 1, we probably run several dozen, but for other people as well, for mouse brain and a variety of different tissues.
Okay. Have you begun looking at how patient resections compare to pre-RX biopsies? I believe this is for Mark.
Yep, we've been looking at that. Is sort of presented the challenge for us is how do we quantitatively talk about differences in cell organization between a needle core biopsy and a resection specimen. But yeah, that's absolutely looking at dynamic changes in response to treatment is something we're very interested in.
Perfect. And next, how many biopsies can you get on a single strand size MERSCOPE slide? What size biopsies are these? Have you tried putting resection and biopsies on the same slide? This will be Mark? Yeah.
I guess for us, but Alex may have more experience in-house. So we have a very talented histologist, Desiree, who has been putting various things on there. As I mentioned, we've done TMAs. The needle core biopsies, you can probably fit about 4 or 5 of them. If they're all in the block, it's easy because it's one section. But if you're trying to put cores from different blocks onto the same disc, it gets a little bit tricky, but I think it's very possible to do. And obviously with a ultra slide, you can fit that many more. But yeah, the idea of putting the biopsy and the resection specimen on the same exact slide so you can run the same chemistry at the same time, the advantage of that hasn't escaped us for sure.
Okay. The next we have from Hisu Song. Is FFPE quality experiment with 4033 blocks performed on surgical resection FFPE samples mainly? Or does that include needle biopsy samples as well?
Yeah, it's a mixture. It's a mixture, just like we did for this experiment. It's a mixture of biopsies and resections. And really I think in our experience, the determinant is from where it comes from, what laboratory it's processed in. Age matters a little bit, but you know, the samples that we did here were probably 4 to 5 years old in age.
Okay, and I guess this would be our last question there. Are there any insights you're starting to see from the spatial analysis of neighborhoods?
Yeah, I mean, that's our hope. We see things are changing. We know that they are changing. We're trying to understand why they're changing, what that means biologically. That's another topic for the grant, for sure.
Okay, we have a little bit time for more questions if you have any more. Yes, I wonder if you see any difference in RNA quality between needle biopsy samples and surgical resection samples. That's from Hee-Soo Song.
Yeah, I think that's what I just answered. I think it's variable. There's no consistent difference there.
Okay, perfect. Thank you so much, Mark. And I think we are at the end of our webinar. Thank you so much to everyone. So it was a great set of presentation and Q&A session. Thank you very much to all the presenters and attendees for joining us. Just a final note, access to this recording will be emailed over to your registered email address within 48 hours after the event. And should you have any further questions or require any assistance, please contact Hayley Lim, who has been in touch with you throughout your time with us. Lastly, a very big thank you to Vizgen and all the presenters and Anjana Narayanan for working with us on this. I wish everyone a good day or evening and hope to see you again in our next event. Goodbye, everyone. Thank you, Alex. Thank you, Mark. Bye-bye.
Thank you, community. Thank you, Global Engage.